So I'd first like to thank Steve for the invitation to speak, as well as Kate Spore for doing logistics in the background to allow myself and the 4 superstar undergrads to speak today. So I'll be talking about synthetic biology research, and the main goal from this talk will be hopefully letting you know what I mean by synthetic biology. So briefly, the outline of what I'd like to cover today, although I'm not wedded to this outline. Before I start, I'd like to say if you have any questions, please feel free to interrupt me and ask. You'll be doing me a big favor. It's often hard to take this very high-level approach for giving a talk, and I'm new at this, so you'll be helping me out by interrupting me and asking questions as we go. And I'll adapt the talk accordingly to your questions. So first I thought to cover what and why do synthetic biology. So what is synthetic biology and why do it? And I think it's really easiest to understand when you look at examples of current synthetic biology applications. So I'll cover some of those, and then we'll transition into this undergraduate competition, IGEM, stands for International Genetically Engineered Machines Competition, which is a fancy word for synthetic biology competition. I'll give some example projects that undergrads have done in the past to give you an idea of how exciting it is that undergrads can really drive these projects themselves, get their hands wet, and do the research by themselves with modest advising from older people like myself. But it's really the undergrads that push that program. And that's why the undergrads will be talking about next what their project was, to give you a flavor for what their experience was. And then we'll definitely, at the end, the most important part of the talk will be answering questions you may have. So we'll make sure that we have time for doing that. Okay, so what is synthetic biology? Synthetic biology's aim is to harness cells for useful applications. So the first question is, why cells? Why would we want to harness them? The first reason is that they're capable of amazing behaviors. So this is a movie of a neutrophil, which is part of our immune system. Their job is to sense and then attack invaders. So in this case, a small bacterium. So somehow that neutrophil is able to sense that bacterium and where that bacterium is swimming. And it's so good at that it's able to even overtake the bacterium. Let's see if I can say that again. I'm amazed every time I see this video. So these round spherical objects are red blood cells. So you can see that the neutrophil doesn't care about the red blood cells. It's not sensing those, or at least it's not responding to those. Its motility or movement is specific only to that bacterium. So you imagine that very sophisticated computation must be occurring in that cell to enable it to be able to specifically sense that bacterium and respond accordingly. So we like as scientists to understand how that's accomplished, and then as engineers, we like to mimic that behavior. There are many other enviable properties or abilities that cells have that we'd also like to harness. So we just went over that neutrophil movement, which has amazing information processing that we still don't understand all the details for how that's accomplished, but we're beginning to understand. So we'd like to further study that, as well as other sensing. You imagine that cells in your body need to be able to respond to what other cells in your body are doing, or microbes, single-cell organisms, have to rapidly adapt to their environment as their environment is changing. There's a remarkable ability to both sense those inputs and then do signal processing or computation to decide how we can respond to that environmental cue or input, and then what we call actuate or execute some response to that input. So we'd like to be able to mimic that behavior or even harness that. Also, cells, as you're probably aware, are capable of metabolizing different sugars, different inputs. So by metabolism, I mean the ability to catalyze reactions. So you're probably familiar from your organic chemistry classes in the past that there's a whole science towards making materials, that we've now figured out how to do certain reactions. Well, a cell can do this with extremely high precision. And there's many things that the cell can do, chemistry-wise, that we can't come close to at the bench with organic chemistry. And that can be in two different forms: anabolic, or the scientific term, making stuff. You're probably familiar with anabolic steroids in the news for sports, which is basically making lots of muscle. But we could also use anabolic pathways to make molecules of interest, such as biofuels, so sustainable production of molecules we can put into our gas tanks. Commodity chemicals, such as polymers that go into making carpet or tiles or synthetic wood products, pretty much almost everything in this room, the seats, vinyl. Imagine if we could use cells to make those materials, so that we're not using fossil fuels as it's currently done, but we could use green plant material as our feedstock, feed those to our cells, and have the cells do this complex chemistry inside the cell to produce our molecules of interest. That would be a much more environmentally friendly way of doing of synthesizing these molecules. Also, the opposite of anabolic pathways are catabolic pathways, breaking down stuff. So this would be like breaking down pollutants. So you might have a pollutant in the environment, and you engineer a cell to pick up that pollutant, do chemistry inside the cell that converts that pollutant into some molecule that's benign, or better yet, a molecule that we could actually use to make a carpet or to drive our car, etc. Or biomass. Can we grow plant material like, let's say, switchgrass, and then convert that plant material into a fuel that we can put in our car and burn? The second reason we like to study cells and harness cells' abilities is that they have a remarkable ability to self-replicate. It's a property of living material that you can make many copies of itself. So if you engineer your cell to be able to, let's say, make a biofuel, you'd have that single cell, but then you could quickly make many, many copies of that factory, that cell, to do the reaction that you desire. So you can rapidly amplify your systems. Okay, so I kind of danced around this, but there's two main goals of synthetic biology. So the first is, as we've been discussing, being able to perform useful functions or applications, such as making the biofuel. And that's a huge part of synthetic biology. The second aim of synthetic biology is to also understand better how cells naturally work. So we often quote this famous line from Richard Feynman: "What I cannot build, I cannot understand." So we really truly understand how a cell is working, then we should be able to reconstitute that and build it back up from the ground up. And if we can't do that, then we probably don't really understand. It's probably missing some component that's important for function. So I'd like to draw the analogy of doing synthetic biology, or you may have heard the terms genetic engineering, to a computer. So you're probably familiar in a computer that you have to write the program that you want the computer to execute in a language the computer can understand. And then that will be coded as binary zeros and ones. And the computer can then actuate or execute that program. So much in the same way, synthetic biologists have to put in the program they want to be run in a language the cell can understand, and we code that in DNA bases, so A's, G's, C's, and T's. And then the cell recognizes that coding and that program to then execute it. So living cells have their natural programming all encoded in these A's, G's, C's, and T's, which we call the genome. So you probably are very familiar with the Sequencing the Human Genome project that happened about a decade ago. So that simply means reading all those A's, C's, G's, and T's to understand to see what the blueprint of the cell is. Now there's a big jump from knowing what the blueprint is to being able to read all that programming. There's a lot of work that still needs to be done on that, but it's a very exciting ability now that we have that's fairly recent, just a decade old, to be able to know what that blueprint is. And that has expanded to many other organisms besides just our own. So now we'd like to take those blueprints and then go the opposite direction and put in our own programs, again encoded in the DNA bases, so that we can then boot up that programming in our cells of interest. So how's the genome read then? So this is called central dogma, and I've oversimplified it here, but I think it'll at least give you a feel for what's going on in the cell. So the cell will have its DNA, drawn here in blue, and then there's machinery in the cell that reads that DNA and does what's called transcription. It converts the DNA into RNA, and then that RNA is then converted into protein by a process called translation. So it's not important to remember all these details. I really just want you to get that DNA is then read to produce protein, and proteins, for the most part, are the machinery that actually does the work for the sensing and signaling metabolism that we talked about. So as I said, proteins do most of the work. They're able to catalyze chemistry in the cell that far exceeds what we can do as organic chemists at the bench. So we really like to harness this catalytic ability of enzymes. The best of these enzymes can catalyze reactions up to 10 to the 7th per second. So you think about that. 10 to the 7th reactions are happening every second in the cell with these best enzymes. Also, signaling. So to produce that directed motility, that movement of the neutrophil I showed you in that first movie, somehow the cell is able to sense an environmental input, and that black box somehow is proteins. So you have receptors at the membrane of the cell that separates the outside from the inside, and that receptor is able to bind environmental input, and when it binds, and only when it binds, does it produce a signal that it's able to relay from protein to protein until ultimately it produces the appropriate cell response. Often that response is expression of other proteins. So you might want to produce protein that catalyzes a new reaction that will give you resistance to a molecule that's in your environment. Or so to do that, then many proteins are involved in being able to read the DNA and be able to DNA or RNA and produce other proteins to do still more responses. So the only basic thing I wanted to get across from this slide is that the DNA encodes for protein, and then protein can do a wide variety of different tasks, or different behaviors in the cell.
Question?
Sure.
Well, in the example you just showed us, we're not creating new proteins. We want the thing to move in the right direction to catch its prey.
Right.
So that's very different.
Right. So in that example, there's signaling that controls our what's called cytoskeleton, which is other proteins that give rigidity to the cell, and the cytoskeletal proteins themselves can polymerize, so it can form long filaments. By that process of polymerization, it pushes the cell forward. And that's a dramatic oversimplification because there's a lot of other factors that are involved in that motility, which is why I chose it as an example, because it's so sophisticated. But much of the work is done by these other proteins that can be either soluble, floating around, or polymerized to form force.
After that first series, after the cell captures that, was it a bacteria? Whatever it was, the proteins.
Yes.
What then?
So, if you notice at the end, the cell didn't have any motility at the very end. It just kind of hangs out for a little bit. And then eventually, another bacterium would come across its path. It would sense that and start moving towards it. So I think there's also, in the sophistication, it's able to lock on that one bacterium and follow it. And it's ignoring everything else. So once it engulfs that bacterium, it doesn't really know what to do for a little bit. It resets. So it would depolymerize those filaments and it would go back to its basal state, its normal state, until it gets excited again by another target.
Whatever the material is that it just captured, does it just stay there?
Oh, what's the fate of that bacteria?
Do the proteins do something to it?
Yeah, so once it's engulfed, it kills that bacterium. It breaks it apart. So other proteins are involved in that as well.
That's catabolic.
Yes, catabolic. A lot of catabolism is done. Exactly. Okay, so now we want to be able to boot up our own programming. So we make our programs. That's what the synthetic biologists do at the bench, and we make our DNA that's in the right language, and then somehow we put those into these cells, and the cell has all the machinery that it can boot up that program and execute, hopefully, our desired function. So I'll give you a brief flavor. These details aren't critical to understand, but I'll give you a brief flavor of how this is actually done at the bench. So we'll have our microbe. In this case, I have a cartoon of an E. coli cell, which I'll go into a little more detail in a minute. And then we have our DNA, a circular piece of DNA that we call plasmid. So the plasmid has all the machinery that it requires to be able to make several copies of itself inside the cell, and has the programs to express the proteins that we'll encode in our genes or our DNA on the plasmid. So we make the plasmid at the bench, and then we insert it into our bacterium, it's E. coli, by a process called transformation. So basically, I just want to get across that we can make our DNA, and then we can put it into the bacterium. How we make that piece of DNA is we'll start with the plasmid. We have proteins, enzymes that very specifically will cut certain sequences. So we can cut only where we want to cut the plasmid. And then this cute example, we want to teach the cell, tell the cell, turn red. So we're going to put in another piece of DNA that encodes for a protein called red fluorescent protein. So if this gene is expressed, then it will produce red fluorescence. So we cut that gene that encodes for red fluorescence with the same enzyme to make the same ends DNA cuts that our plasmid will have. So that using that, then we can then put that red fluorescent protein gene into our plasmid. And we use another enzyme that sews it all together to make a now continuous circle piece. It's all enclosed as one contiguous piece of DNA that now encodes for our red fluorescent protein. So we transform that plasmid into our bacterium. It will express our red fluorescent protein. This is the structure of that red fluorescent protein. And then with time, the cell will get more and more red. So you can actually see colonies of these cells. So each of these circles is many E. coli cells growing right next to each other. So each colony was a single cell that got onto our plate here, and then they multiplied until we could visibly see them as a circle. So then synthetic biologists will take that genetic engineering, the ability to be able to express red fluorescent protein, and now we add in sophistication. So it's not important to understand what this cartoon is doing, the details of it. I just included it to show that we can now we can add in other elements to make this expression more stable. So this circuit is called a toggle switch, which behaves just like the light switch, where it can be either off or on, but you'll never get intermediate. That has no dimmer. It won't be half on or half off. It will either be all the way on or all the way off. And it accomplishes this just by simply making repressors that will turn off expression of another repressor. So if you have expression of one repressor, that'll turn off expression of the other, so on. So you can induce RFP be all the way on or all the way off by adding this genetic circuit. So again, it's not important to understand the details of this. I just want to give you a flavor of what you can start building into the cell to get the desired behaviors. And I included this one particularly because in 2000, Gardner and Collins made this toggle switch, and it was really one of the first efforts of synthetic biology. So this is when we really started realizing around 2000 that we could take this approach forward. So I would say synthetic biology was really born around 2000 with this first demonstration. But to build these sophisticated circuits, you need to have a lot of parts. So one of the efforts that was started early on by MIT was to make a registry of standard biological parts. And this is a website that you can go to, and you can actually order many different types of parts. So you have parts for making chemicals, and for biosafety, for controlling whether your cell lives or dies, et cetera, et cetera. So many different types of parts. And this is important because just like with the computer, you want to be able to go to Radio Shack and be able to pull out parts off the shelf and know that any one part can function with another part, so you can combine them together like Legos. If you can't have this ability like a Mac computer, where the Mac's great if it works and you have a Mac store, but if it breaks, you have to take it to the Mac store. You can't just go to Radio Shack and take a part off the shelf and plug it in. You really, as a field, we wanted to be able to have as much standardization as possible so that we could just take parts from what other people have done and plug it in with other parts that we have made. So we don't always know beforehand what the final destination of each of these parts is going to be. So maybe another way of thinking about this is the example of a screw, where you can make screws with different heights of ridges or different lengths between these ridges, and the screw would work, but it wouldn't work with other prefabricated materials. You really ideally want to be able to buy a screw from a hardware store in San Francisco and be able to use it with a nut or that you bought in Boston, for example. So you want to have confidence that each of these parts will go together. Even though when they are both manufactured, the manufacturers didn't know that you were going to put these two parts together necessarily. So to do that, there was an international standard for the thread size and pitch diameter that was built, so that no matter what device you're building, these screws will work with those prefabricated materials. So we'd like our biological parts to be built the same way. So we, as much as possible, can put any one part together with any other part and still expect them to function together. Okay, so I gave you that cute example of red fluorescent protein production. obviously that's not going to save the world doing that application. We'd like to now apply the same principles to and add some sophistication. Let's take the example here of making a small molecule. So again, this is a somewhat simple example, but it'll give you the idea. So in this case, we built our 3-enzyme metabolic pathway using a language the cell will understand. So that is adding a promoter, which is a part that the cell will recognize, okay, I'm going to express any sequence of DNA that's immediately downstream. And in this case, that would be an enzyme called 8OB. We do the same thing for another enzyme called HMGS, and then the same thing for a third enzyme called HMGR. So now we put this metabolic pathway at the DNA level in a format, a language that the cell will understand, so it can produce those enzymes. So now we have an enzyme, 8OB enzyme, HMGS enzyme, and HMGR enzyme. We can feed the cell this small molecule, and this enzyme will catalyze the reaction to convert this. It actually will take 2 of these and make this molecule, which will then be recognized by this enzyme to make this molecule, which then will finally be recognized by this enzyme to make our final product. So this works, but inevitably we run into some problems. So in this case, it works, but we have toxicity because we're building up a lot of this intermediate. So one solution to this problem would be to build in more regulation. Or simply, we can use a stronger promoter part. So this promoter will produce more enzyme than these other promoters will. So now we have many copies of this enzyme. So we will no longer accumulate this intermediate, but will produce lots of final products. So we took out that bottleneck and relieved the toxicity while also producing more of our final product. So problems like this toxicity often arise because property of synthetic biology is taking enzymes that the cell never had seen before and making molecules that cell, our production host, had never made before. So it hasn't evolved to deal with these high concentrations of each of these molecules. So there's a lot of synthetic biology applications that go into making pathways like this work better. And this is a simple 3-enzyme pathway, but you can imagine scaling that to much larger pathways, like 10, 15 enzyme pathways. The more sophisticated your final molecule is, the more enzymes you're likely to need to catalyze those reactions. Okay, so I've been using the example of E. coli as our production host for this talk. E. coli is probably still the most often used organism. There's many advantages to it. So it can be grown both in the presence of oxygen, aerobic, or in the absence of oxygen, called anaerobic. That can be advantageous because we grow a lot of these cells in a really big fermenter, a fermenter bigger than this room, and we grow them to a very high density. So there's many cells in that fermenter. Then it's actually hard to get enough oxygen in to that fermenter for the cells to be aerobic. So any process that's going to be grown at a really large scale has to be able to grow in the absence of oxygen. So E. coli has that ability. It's very abundant in our large intestines. So you may have be familiar with E. coli. Actually, the wild type strains can be toxic and can create large problems. The strains we use in the lab have been neutered of all these toxic virulent factors, so it's safe. It's actually a BSL-1, which is biosafety level 1 organism, the most safe to work with. So it's nice in the laboratory setting for that as well. It does have, and as great engineering tools. So being able to get DNA into the cell, that's been worked out. That's very easy to do. We have a lot of parts the most studied, most used organism. So those are its advantages. One of the other organisms, probably the second most often used organism that the IGEM students will talk to you a bit about is yeast, baker's yeast, Saccharomyces cerevisiae. So it also has many advantages. It's a simple single-celled organism like E. coli, but in this case, it's actually a eukaryote, which is more like our cells, which means it has organelles. So that's what this picture on the right is showing. These are supposed to be different organelles. It also can be grown aerobically and anaerobically. One of its advantages is actually it's very resistant, naturally, it seems, to viruses, which you can imagine you're growing a large fermenter, a large-scale growth of your organism to make, let's say, a biofuel. You want to make sure you're not going to lose that whole fermenter to some pathogen for that microbe, some virus for that microbe. The yeast doesn't naturally have nearly as many viruses that attack it as E. coli does. It's also hardy or robust to low pH and other chemicals, which can be advantageous. You can grow at a low pH, and a lot of the bacterial contaminants that might grow in your fermenter cannot grow at that low pH. So you can keep it relatively clean of contamination. Okay, so synthetic biology has the characteristics, I think you probably have appreciated by now, of creating approaches for facilitating the programming in living cells. So we want to be able to program more, bigger and better. It has a focus on regulation, so we can control when something's made, where it's made, how much is made. And to do this, often we're building multi-gene circuits, so multiple pieces of DNA put together. And we try to build those in a modular way, so think Lego, so we can put any two pieces of DNA together and still expect it to function. And to do this, we're obviously exploiting new DNA synthesis methods. So we're getting much better at making these DNA parts and being able to assemble them together. And we're also employing parts that we get from a variety of different sources. So different organisms get enzymes from different places. So I'm going to give you some quick examples of projects. So one that you may have heard of in the news that came from right here at Berkeley, Jay Kiesling's lab, was the ability to synthesize, and they actually used E. coli. They separately used E. coli and yeast. Yeast is actually the one that's being commercialized now, to produce the anti-malarial therapeutic artemisinin. So this is a wonder drug that you can treat malaria, and it's naturally in a plant, Artemisia annua, that's only grown in China. So it's not really a problem of producing it per se, but it's a problem that it's only produced in this one area of the world, and that it's subject to different weather patterns. So a whole crop could be wiped out if there's a really bad weather pattern that year. And the real emphasis is being able to produce this molecule very cheaply, because most of the patients will be from third world countries, and they can't afford any therapeutic that's more than $1 or so. So the idea was use a microbe, put in the enzymes that will catalyze reactions, ultimately produce that drug, and then you can scale up and grow fermenters that's large with those microchemical factors. To be able to do that, you obviously need to find the right enzymes to catalyze each of these reactions. You need to get them to work in your yeast cell. You need to fine-tune the expression levels as we discussed earlier. And in some cases, you may have enzymes that have very poor activities. You need to be able to make those enzymes have higher turnover activities, with more reactions per second. So this was highly successful. They're able to make this, and it's actually going to third world countries this year for very, very cheap cost. Another nice thing about it is the way the pathway was made. these enzymes, they can actually go from this antimalarial therapeutic, artemisinin. They can take out 2 enzymes and replace it with one other enzyme, and now they make a molecule that looks like a good fuel that you could put in your car, farnesene. So the spin-off company of JS Lab, Amyris, is actually making this molecule so it can be a jet fuel, biodiesel. So another nice example is this thought of using bacteria to seek out and then destroy tumors in the body. So we've already seen that cells can be naturally made to be very good at sensing and targeting other cells in the body. Maybe we can do that with a bacterium. Make it sense a tumor, and only when it senses that it's inside the tumor does it produce a payload that will kill that tumor cell. So there's extreme challenges with doing that. I don't want to make that sound like this is a application that's almost ready to be unveiled. This will take a long time to be able to accomplish. You can probably appreciate that you need to be able to build the bacteria to first survive in the bloodstream. We have a great immune system for bacteria because they're usually pathogens for us. So if you try to inject a lab strain of E. coli in your blood right now, it would be catabolized in a matter of seconds to minutes. But you also, once you get it to survive, you also need to engineer it to do no harm, to not cause sepsis in the body. So that also would require a good deal of engineering. And some progress has been made on these fronts, but we're still a long ways away. So this is work being done by Chris Anderson at Markham here at Berkeley and Chris Boyd at MIT.
I'm sorry, the bacteria, would it then stay there if there was no recurrence of the tumor?
At least the original design, I thought, would be that once it's killed the tumor, then it would self-destruct, so that you wouldn't have loose bacteria floating around. So you can think along the lines of making probiotics. So you're probably used to eating yogurt as a probiotic, but you could start making those probiotics, maybe produce vitamins or produce some protein that if you have a disease, you're lacking one protein. Because you're lacking a protein, they can produce that protein. So there might be some therapeutic applications.
How many years off would the tumor-killing bacteria be?
I think tumor killing is probably the most ambitious application. That's probably far enough away that it's hard to guess, like 30, 40 years at least. In probiotics for certain diseases, especially intestinal diseases, that might not be as far off. It might be like decades ago.
One question.
Sure.
Would you mind repeating the question that was asked?
Sure. So the last, did you specifically this last question? Or
No, just as a general rule.
Yes, I'll do it. Thank you. Okay, so engineering these complex behaviors is, I made it sound simple on the slides, but it's actually extremely complex. Making that artemisinin example, that has taken hundreds of people years. So the company Amyris that did this had about 100 employees at the time working on this problem. It took them a few years to do this. We'd like to be able to do it much faster, cheaper, easier. So I won't spend time on this, but just to tell you that one of the efforts is in both standardizing, as I said earlier, and then also using liquid handling robots that can do the benchwork for us, so we don't have to rely on our undergraduates to do it. And also, we have a very poor understanding of how the cell works. This is a new field, as I said, a little bit over 10 years old. So right now, we go through this cycle many, many times and learn each time we go through. So we make our parts, then we put those parts together to make our program, then we test how well they work, and then they almost always fail. So then we have to redesign and go through the cycle again. So briefly, I want to give you a little bit of an introduction to this undergraduate competition called iGEM. So iGEM was created around the question, can simple biological systems be built from standard interchangeable parts and operated in living cells? Or is biology too complex to have hope of accomplishing this? So you obviously know what my feeling is from the first part of the talk, but this was a real question, especially about a decade ago when we were embarking. There were a lot of naysayers for whether this could be accomplished, and it's still a Herculean task to get even simple things that look simple on paper when you draw it out to function the way you expect them to function in the cell. Because there's a lot of things going on in the cell, a lot of chemistry, a lot of signaling in the cell, and we only have scratched the surface of understanding all of those processes. So iGEM has really grown over the years. So remember that first paper I showed you is in the 2000s, so that's when we were starting to think about this as a new field. In 2004, there were 5 teams, and now there's over 200 teams, almost 250 teams. So it's rapidly expanded. It's expanded so much that we used to have the whole competition in this one room in the end, and now we don't all fit. So we have to actually have staggered competition. And much of that growth is because it's expanded from beyond just the United States to international. So each of those dots represents a team. Okay, so the process of iGEM is one of the beautiful things about this is it all takes part in one summer and one fall. So our competition takes place in the end of October, beginning of November. So we start in the beginning of summer, and we have to pick a problem that we want to address. Then we do experiments in the lab, and that takes that summer and fall, beginning of fall. And then at the end, you build a website, so you can check out their website by Googling Berkeley iGEM 2012, and you'll get to this page. So that way they advertise what they did, and you can see any other iGEM team from any of those years on the web as well. And then you write a poster. So it's a great experience for the students to be able to take part in designing a project around a problem they're interested in, and see it go all the way from that design of experiment all the way to the website, poster, and a talk at MIT. And one of the other nice things is all these other students, the other teams are doing the same thing, so they enjoy meeting each other and they can actually understand what each other is doing. They really learn a lot from this experience. Also, I wanted to highlight, are there any high school students in the room? All right, great. So there's also now a high school competition that just went online, I believe, last year. So you might want to check that out as well. Okay, so I think I'm running out of time, so I'll just go through these really quick to give you a flavor for what they're able to accomplish in these summer projects. One of the first teams in 2006 that I like is building a arsenic private sensor. So a real problem, and especially in India, is that many wells are inadvertently dug through arsenic veins. So the well water is contaminated with arsenic. And there's about 100 million people in India that are contaminated every year. And about 1 million of those die from cancer every year. So this team from Edinburgh wanted to use E. coli, and they built a pretty sophisticated genetic circuit that would sense all the way down to 5 parts per billion of arsenic, and then execute through the circuitry a response that lowers the pH. So then you could measure that lowered pH and say, okay, this well water is contaminated with arsenic, you shouldn't drink it. In Cambridge in 2009, they had a really fun project they called E-chromide, which they made produce molecules that have different colors. So this, you can imagine hooking into this biosensor in 2006, so they made this biosensor before the E-chromide. You can imagine instead of lowering the pH, you could make one of these colors. So it'd give you even cheaper output that you could use in the third world or in countries like India for detecting whether you have arsenic contamination. So it makes an output that's much cheaper to measure. MIT had a fun one in 2006 where one of the bad things about E. coli is they stink. If you've ever worked with them. So they did a fun project where they produced wintergreen or banana smell. So the bacteria smell good. And then they also deleted one of the proteins that gives bacteria bad smell. Berkeley in 2007 had a project where they thought, what if we could make bacteria cells that you can think of them as just bags of hemoglobin, so a synthetic red blood cell. The team that won this year was Groningen, where they made a circuit in a bacterium that could sense volatile molecules are given off by rotten meat. So they put these bacterium into a capsule with another compartment that has nutrients. So you break that barrier, so the nutrients mix with the bacterium starts growing, and then it'll produce a fluorophore if it's in the presence of that rotting meat volatile molecule. Okay, so I will hand it off to our iGen team to talk a little bit about their projects in this last year.
Okay, so we're not going to be using the microphone, so I just want to check. Can everyone hear me?
Yeah.
Okay, excellent. Okay. So the concepts we've covered so far really go to show that the goal of synthetic biology is to make it easier for us to engineer cells with new capabilities. And when we design a new system using biological systems, we usually like to utilize certain basic features of cells. So we've covered some of these, but I just want to briefly go over them again. Here's a neutrophil example where the feature we might want to use is how the cell moves around in its environment. Another really cool feature of cells is how they're shaped. So many cells can form different shapes, and that allows them to interact differently with the cells in their environment. So here's an example of that. And another really cool example is actually how the intracellular contents of the cells can be organized to form different types of structures. So here we have yeast cells that have a different fluorescent or colored proteins localized, as you can see, to the outer membrane of the cell. So even just from these three examples, we can really start to see that biology gives us many interesting and useful features with which to work. And we realize that if we're engineering a system that utilizes one of these features, one of the best ways to see if that system is actually performing our desired function is to be able to look at those cells directly. And we found that microscopy is actually a very powerful tool that can let us carry out this process or procedure. So a microscope, as you all know, is an instrument that can let us see objects that are much too small to be visualized by the eye. So using the power of microscopy, we can see how internal cellular organization is performed. We can see how different cells form networks with other cells in their vicinity. We can also see how cells may move around in their environment and be caused to move around in a different fashion based on the environmental factors present. So I thought we'd do a simple thought experiment of how we use microscopy to analyze the system that we're trying to engineer. Now, in synthetic biology, we often know the starting point, which is maybe the genes we want to encode in DNA to carry out a particular function. And by extension of knowing the genes, we know which proteins they'll produce. But one of the problems that we sometimes run into is that we don't know how much of each protein we want to express. So the earlier example we covered with the metabolism, where we had expressed certain proteins in some ratio, but we gained toxicity. So we have to go back and add in more protein at a certain time point so that the toxicity could be alleviated. So similarly, if we're designing a new system, we usually want to try out certain settings in different sample types. So let's say we start off with 5 cell types, where each cell type has some different setting of the genes and the proteins being expressed. What we currently do is we take and prepare each of those individual samples separately. we would then visualize each cell type under the microscope, and if we like what we see, we would sequence the DNA. So this essentially acts as a verification method for finding out again which cells we just looked at. And if we like what we see after that, we're happy because the cells are carrying out the function we want. But rarely, if ever, do we find an optimal setting using just 5 cell types. What we actually have to go through is maybe somewhere on the order of 5 million cell types. And as you can probably imagine, if we were to try and go through this exact same workflow of analyzing each of those 5 million cell types one by one, we're not going to be happy anymore. It will take much too long, and it will be much too inefficient. So the problem we're really trying to tackle this summer, and the central question we asked ourselves was, how can we retain the power of microscopy in letting us observe these features, but at the same time not be limited in the time it takes us to analyze our cells?
So one way to go about this is by mixing your set of population of cells on one microscope slide and viewing it under the microscope. So in this population of cells we see, the first one they're all varying by the level of red we see in the nucleus. So the first member of this population we see has very strong reds in the nucleus. A second member may have a weaker shade of red, and a third may have no red at all. So if we find a particular cell that we're interested in, say the one with a medium level of red, there's an inherent flaw. While we see that as the appearance shows that a medium level of red, we don't know the specific genetic manipulations that brought about this appearance. So the inherent difficulty that we try to tackle this summer is the link, that there's no link between the genetic identity of the cell with its physical appearance. So to go about this, we create a system of barcodes. Now this system of barcodes will link the specific genetic identity of the cell to its physical appearance. And to do this, to create these barcodes, we use the unique property of yeast is that they have specific compartments within the cell called organelles. And so to create this barcode, we can vary the number of fluorescent proteins, or colors we see in the cell, and the number of different places these proteins exist, or within the organelles or compartments. And for our purposes, we use 3 different colors, red, green, and blue, and we use 4 different organelles: the cell periphery, the outside of the cell, the nucleus, the vacuolar membrane, and actin. So by varying different combinations of these organelles with different combinations of these colors, we can create an exponential increase in the number of barcodes that we can create. So now I'm showing you a cartoon example of how these barcodes appear, but how do they look in real cells? So here are 4 examples out of the multitudes of barcodes we created. These barcodes are called mycodes, or microscopic barcodes. And as you can see, they each have a unique visual identity that allows them to be distinguished from each other under a microscope based on the combination of colors and where the spatial organization, or where these colors exist in the cell. So again, at this point, we've created this visual fluorescent barcode that's linking this specific genetic identity with the physical appearance that we're interested with the cell.
So now that you guys have an idea of what microcellular barcodes look like, let's go back to a very quick example of the cartoon version. Say you have a microscope image of several million cells, right? You want to identify all of them, and you start out not knowing which cell corresponds to contains what genetic material you put in. Right? So you look at this slide and you have all these colors. How exactly are we going to go through all of these cells, in a reasonable amount of time, and identify each barcode it has? Right, so the first idea you might think of is, just go through it by eye. Scientists would go through all of these images and identify each individual barcode, and then, from that barcode, be able to tell what genetic material that cell has. But of course, as you can see, that this would take much too long and much too laborious. Right. So as the second part of our project, we turn to kind of the computer automatable method, in which we develop software to kind of act as a barcode scanner for these cellular barcodes. Right? So instead of identifying, looking at the colors by eye, we would feed images of the cells into a computer and have the computer do the scanning for us. So essentially what you have is, a series of images of cells, and you feed them into a computer, and we've written software that actually kind of detects in the image parts of the cell that are fluorescing different colors. reconstruct what the cellular barcode has and determine what that genetic material is. So essentially, as the second part of our project, we've developed kind of a barcode scanner to use for these microbes.
We first tested our microcodes out with 2 different types of cells. Cells that show that show red everywhere in the cell, and cells that show red only in a certain organelle. We linked them to microcodes. And so if the cell showed red in only one organelle, it had a blue membrane and green nuclei microcode. And if they showed red everywhere, they showed a green membrane and blue nuclei microcode. And so you can see, without looking at the slide on the right, you can tell which microcode the cells are. And while this is it doesn't show very much in this with just 2 types of cells, but if we scale it up to more cells, this type of microcoding becomes very useful, as you will see in my next slide. So during our project, we began a collaboration with a lab at MIT, the Keating Lab. And what they do is they computationally design genes on the computer. And when we put these genes in our cells, if they interact with each other, they will give us red in only one organelle. However, if they don't interact, they will show red everywhere. And so we put these genes into our cells, and we have a lot of different genes, and we mix and match them to get just test out all the different combinations of the genes. And with each combination of gene, we attach a specific microcode. And so you can see when we put them on the slide, we have lots of cells that don't have interacting genes. So you can see all the cells with the red everywhere. But we do see a couple, 2 cells here at least, that have red only in one organelle. And those are the genes those are the cells that we are interested in because they show that the genes interacted. And so we can go back to the microcode, and from that microcode we can figure out what genes made that interaction. So in summary, microcodes allowed us to see from the physical output on the right what genes or genetic information caused that output. We'd like to thank, so during the summer, a lot of people helped us out with our project. We'd like to thank Agilent Technologies for the financial support, Sinberg and the SynBio Institute for their administrative help, the Dueber Lab for supporting us and answering all our questions, and the Keating Lab for their collaboration. John Dueber and us on the team would love to answer any questions you have. Thank you for your time.
So I want to really quickly make just a couple of acknowledgments as well. Will Deloche was the graduate student mentor for the team. He deserves a lot of credit for the advising. Terry Johnson also served as an advisor, especially in producing the presentation, helping them with their presentation at the end, and for funding Agilent Technologies and NSF for financing the team, and Kate Sport for the logistics and organization that goes behind the scenes for making one of these teams work. With that, I would like to take any of your remaining questions.
This is about time scales. What you showed us at the beginning of the talk, but you didn't tell us, was that real time or had you sped time?
Okay, the question is about timescales for that first movie. Is it in real time or is it sped up? It's sped up, and I used to know exactly how much, but I can't remember.
Order of magnitude.
It's at least, I would say it's probably about 10x sped up, but I don't know the exact number.
I'm trying to understand the barcode analogy. So I think of a barcode as pretty flat, right? And you sort of flash or QR code. How does this one work if you've got layers? You've got so many layers of sound.
It's a great question. So the question was about the analogy of a barcode. How do you measure the barcode when the cell is three-dimensional? And instead of being flat. So for our measurements that they were doing just simple wide field fluorescence, which doesn't take into account, which will take the whole image. You actually can do an image that would make this barcode much higher resolution. That's a microscopy technique called confocal microscopy that is able to just take a slice, and then you have all that information. It's called Z-stacking. You take several images of slicing as it goes up and down, and then you can reconstitute exactly what the cell looks like in 3 dimensions. So that will tell you a very clean, high-resolution picture. We weren't doing that because that takes more time, but that would be a way of getting higher images and going to more organelles, so you can get a more sophisticated barcode, higher numbers in your library. What they were doing, you're right, that it's 3 dimensions, so we were sacrificing some of that resolution because we can still see it by eye and by Thomas's algorithm. But yeah, the analogy is simply meant to be that analogy, not the details, but the ability to link a program of DNA that we're introducing with some visual code that we can say, that cell has this gene, or has that program, that cell has this other program. In the back.
When you're using enzymes, how do you know which enzyme to use to catalyze a specific reaction?
Okay, so the question is, when using enzymes, how do you know what's the right enzyme to use for your pathway, to catalyze your reaction, right? That's a great question. There's actually a lot of work being done in characterizing enzymes by scientists, not just synthetic biologists, but scientists. And as people are going to the oceans and sequencing lots of DNA from different small single-cell organisms in the ocean, and going to tropical rainforests, and there's a lot of biodiversity of organisms there. So we're rapidly increasing the number of enzymes in our database as well. And I would say there's a huge backlog for characterizing all of them. Right now, most of them are done by a work called homology, saying the sequence of this enzyme looks similar to this other enzyme, and we've already measured what this other enzyme does. So a lot of it's done by inference, of guessing, because this looks a lot like this other enzyme. It probably does that same reaction. Whether it recognizes the same substrate, the same starting molecule, that's another question as well. And I think a lot more characterization needs to be done. What has been done so far is all cataloged in websites. There's one called Brenda, Renda, like the woman's name. And you can see many enzymes characterized for what reaction it will catalyze. So we do put it, when we characterize it, we do put it in a site where everyone can look it up. But there's a lot of enzymes out there we don't know what they do. So it's a great question. Any question?
John, my question relates to the search for life out there. And you mentioned at the beginning of your talk that to do synthetic biology, you need to get your hands wet. It's the wetness element, the presence of liquid water as an essential ingredient for life. Is it possible, from your perspective or your student's perspective, that life could exist in the absence of liquid water?
So the question is, everything we talked about, water is necessary component. Can you envision a world without water where life still exists? It sounds like you really need to go to the next talk. It sounds like that. The talk you yeah, the way life works on Earth, no. It would have to be a life form that uses completely different chemistry than what we use. I'm not it's out of my area, so I don't know if that's infeasible that it could have different chemistry. But the way life works here, you definitely need water. low temperatures, absence of light. So the light's found a way to work in many different conditions. To my knowledge, you need at least a little water to do some of the chemistry that's required. In the back of the red.
The RGM project is very interesting and potentially would have an important diagnostic application. It is now known that there are many tumor markers. So have you applied, say, tumor markers in staining the cells and then trying to identify the cells in cytology material?
So the question is, it seems like the barcoding could have diagnostic implications, especially for tumors where you get many different cell, phenotype of cell. Basically, the genetics are going amok. Could we envision using dyes along with our barcoding that would be specific for certain types of tumors? Is that right? So we have thought about, we have not done anything along those lines. We've only worked in yeast so far, but we have thought about going to different organisms, especially human cells, and we've actually also specifically thought about tumor masses, so we could see what is the identity of each cell in that tumor mass and what's the niche that it occupies spatially in that tumor cell. We've only been thinking about it, we haven't done anything towards that. The dye question is actually an interesting one as well. We started thinking that you could also use dyes to increase the complexity of the barcode and the confidence of the barcoding. You can use dyes actually for individual organelles as well, so that you could have more confidence. This is indeed an organelle, I think it is. And you could expand the number of organelles that way. So you could use organelles that have somewhat overlapping images produced, but still have the confidence that this is indeed organelle A and this other one's organelle B. So we started thinking about using stains from that application. We haven't really started thought about using it for tumor identification. That's a really good suggestion.
Have you thought about making multicellular organisms? Because you've mostly been talking about single cells.
So the question is, have we thought about using this strategy for multicellular organisms? So far, we've only applied it to unicellular. But yes, we have thought about it. We think that would be really interesting to see how, again, spatially, what the different identity of cells are that come together and what the arrangement is. We haven't done that, but you can imagine it being interesting for looking at differentiation as one cell changes into a different morphology. You could see what genes were involved in that change to that morphology. So we could express different proteins at different levels and see which ones made it to the morphology we desired, and we could track that with the barcoding. It's more something we're daydreaming about right now. Probably not going to do that anytime real soon, but I think it is a suggestion that we have thought about and think would be really neat. Really neat.
Is the barcoding specific to color only or intensity of the fluorescence?
So I'll repeat the question so that the mic will let her answer. So the question is, so far we've only talked about using the identity of color as a signal for the barcoding. Have we thought about using different intensities of signal for the barcoding?
Yeah, so far in order to get a barcode set that could help us for our experiments, it was enough to use location and color. But yes, intensity is actually another way to expand the possible set of barcodes you can make. That requires some additional effort on the computational front, but it's definitely something that we feel is feasible to do if we ever need to make more barcodes. Yeah,
so one of our students that's not here, I saw in the picture, our team's actually 7 students. 3 weren't able to make it. One of the other students was doing work on using the same colors that we showed here but a version that is photoactivatable by UV light. So you only get that light up if you first shine it with UV light. So you can take an image, see where all your colors are, then shine UV light and see what additional places did that color light up. And that's another way of getting increased complexity in the barcode.
How much additional effort is it to put in the color coding? Like, isn't that to be done while also doing the genetic engineering?
Yeah, that's a great question. So the question is, how difficult is it to make these barcodes? Can it become just something you routinely do whenever you're making your programming that you're interested in making for your desired application? Just link that automatically to a barcode. So Robert was the one who made the biggest library of barcodes, so I'll let him answer that.
So it does take additional effort, but it's done at the same step as when you put in the genetic information. So you put in both the genetic information and the barcode, but you just make them at the same time and put them in together. So it doesn't take an extra step. It just takes something you do at the same time.
So Robert's modestly saying that you do it in parallel, so it takes extra effort on Robert's part, but it doesn't take extra time. He values time more than anything.
Well, if there are no more questions, again, please come back next month. I'd like to thank Chris Klein, who's been doing our videos for us here. That should be up on the web during the next few days if you want to share that around. And again, thanks to Kate Spohr and Stenberg, and thanks to Professor Dueber and the team at Undergrads here. Thanks.
So I'd first like to thank Steve for the invitation to speak, as well as Kate Spohr for doing a lot of logistics in the background to allow myself and the 4 superstar undergrads to speak today. So I'll be talking about synthetic biology research, and the main goal from this talk will be hopefully letting you know what I mean by synthetic biology. So briefly, the outline of what I'd like to cover today, although I'm not wedded to this outline. Before I start, I'd like to say if you have any questions, please feel free to interrupt me and ask. You'll be doing me a big favor. It's often hard to take this very high-level approach for giving a talk, and I'm new at this, so you'll be helping me out by interrupting me and asking questions as we go. And I'll adapt the talk accordingly to your questions. So first I thought to cover what and why do synthetic biology. So what is synthetic biology and why do it? And I think it's really easiest to understand when you look at examples of current synthetic biology applications. So I'll cover some of those, and then we'll transition into this undergraduate competition, IGEM, stands for International Genetically Engineered Machines Competition, which is a fancy word for synthetic biology competition. I'll give some example projects that undergrads have done in the past to give you an idea of how exciting it is that undergrads can really drive these projects themselves, get their hands wet, and do the research by themselves with modest advisement from older people like myself. There are many other enviable properties or abilities that cells have that we'd also like to harness. So we just went over that neutrophil movement, which has amazing information processing that we still don't understand all the details for how that's accomplished, but we're beginning to understand. So we'd like to further study that, as well as other sensing. You imagine that cells in your body need to be able to respond to what other cells in your body are doing, or microbes, single-cell organisms, have to rapidly adapt to their environment as their environment is changing. There's a remarkable ability to both sense those inputs and then do signal processing or computation to decide how we can respond to that environmental cue or input, and then what we call actuate or execute some response to that input. So we'd like to be able to mimic that behavior or even take harness that. Also, cells, as you're probably aware, are capable of metabolizing different sugars, different inputs. So by metabolism, I mean the ability to catalyze reactions. So you're probably familiar from your organic chemistry classes in the past that there's a whole science towards making materials that we've now figured out how to do certain reactions. Well, a cell can do this with extremely high precision. And there's many things that the cell can do chemistry-wise that we can't come close to at the bench with organic chemistry. And that can be in two different forms, anabolic, or the scientific term. The second reason we like to study cells and harness cells' abilities is that they have a remarkable ability to self-replicate. So that's a property of living material that you can make many copies of itself. So if you engineer your cell to be able to, let's say, make a biofuel, you have that single cell, but then you can quickly make many, many copies of that factory, that cell, to do the reaction that you desire. So you can rapidly amplify your systems. Okay, so I kind of danced around this, but there's two main goals of synthetic biology. So the first is, as we've been discussing, being able to perform useful functions or applications, such as making the biofuel. And that's a huge part of synthetic biology. The second aim of synthetic biology is to also understand better how cells naturally work. So we often quote this famous line from Richard Feynman, "What I cannot build, I cannot understand." So we really truly understand how a cell is working, then we should be able to reconstitute that and build it back up from the ground up. And if we can't do that, then we probably don't really understand. It's probably missing some component that's important for function. So I'd like to draw the analogy of doing synthetic biology, or you may have heard the terms genetic engineering, to a computer. So you're probably familiar with a computer that are making stuff. You're probably familiar with anabolic steroids in the news for sports, which is basically making lots of muscle. But we could also use anabolic pathways to make molecules of interest, such as biofuels, so sustainable production of molecules we can put into our gas tanks. Commodity chemicals, such as polymers that go into making carpet or tiles or synthetic wood products, pretty much almost everything in this room, the seats, vinyl. Imagine if we could use cells to make those materials, so that we're not using fossil fuels as it's currently done, but we could use green plant material as our feedstock, feed those to our cells, and have the cells do this complex chemistry inside the cell to produce our molecules of interest. That would be a much more environmentally friendly way of doing of synthesizing these molecules. Also, the opposite of anabolic pathways are catabolic pathways, breaking down stuff. So this would be like breaking down pollutants. So you might have a pollutant in the environment. Can you engineer a cell to pick up that pollutant, do chemistry inside the cell that converts that pollutant into some molecule that's benign, or better yet, a molecule that we could actually use to make a carpet or to drive our car, etc. Or biomass. Can we grow plant material like, let's say, switchgrass, and then convert that plant material into a fuel that we can put in our car and burn? But it's really the undergrads that push that program. And that's why the undergrads will be talking about next what their project was, to give you a flavor for what their experience was. And then we'll definitely, at the end, the most important part of the talk will be answering questions you may have. So we'll make sure that we have time for doing that. Okay, so what is synthetic biology? synthetic biology's aim is to harness cells for useful applications. So the first question is, why cells? Why would we want to harness them? The first reason is that they're capable of amazing behaviors. So this is a movie of a neutrophil, which is part of our immune system. Their job is to sense and then attack invaders. So in this case, a small bacterium. So somehow that neutrophil is able to sense that bacterium and where that bacterium is swimming. And it's so good at that it's able to even overtake the bacterium. Let's see if I can do that again. I'm amazed every time I see this video. So these round spherical objects are red blood cells. So you can see that the neutrophil doesn't care about the red blood cells. It's not sensing those, or at least it's not responding to those. Its motility or movement is specific only to that bacterium. So you imagine that very sophisticated computation must be occurring in that cell to enable it to be able to specifically sense that bacterium and respond accordingly. So we like as scientists to understand how that's accomplished, and then as engineers, we like to mimic that behavior.